issue is that animals face a new predator, and in previous work we have checked how risk-taking behavior of Anolis lizards affects their survival in the presence of another predatory lizard. And this was done on small experimental islands in Bahamas. Around 300 Anolis sagrei lizards, these ones, were captured in different natural source populations. And then those lizards were uniquely tagged with alphanumeric tags and translocated to 8 experimental islands, 4 islands without predators, so the ones that are marked with yellow circles, and 4 with a predatory curvy tail lizard, this lizard. And the predator islands are marked with green squares. And similar numbers of females and males were introduced to the island, and the density was adjusted to each island size. So this translocation was done in June, and then after 4 months, the islands were visited again, and the lizards were captured to check whether they have survived. So every lizard that was not captured was considered dead. But before the translocation, each lizard went through a behavior assessment test. First, while in a refuge, the lizards were exposed to the predator. And then, after a short acclimation phase, the refuge door was open, and we measured time until the lizard put its head out. And this variable I call head out, and it represents an exploratory behavior of the lizard. And then we measured time that the lizard spent on the ground. So the time when it was full body out of the refuge, but before it got hidden, for example, under the rocks or on the perch. And this variable I call time exposed, and it represents risk-taking behavior because the predatory lizard catches its prey on the ground. And besides the behavior, we also checked some morphological traits. This was done based on X-ray images, and we measured, for example, forelimb length, hind limb length, and body size of the lizard that is represented by SBL, so the snout bend length. So what has been found in this study is that on predation-free islands, lizards that were more exploratory, so they had shorter time to initiation of exploration, so shorter time to put the head out, they survived better. And on predator islands, lizards that were less risky, so the exposed time was shorter, so they spent less time exposed on the ground, had higher chances of survival. And interestingly, this relationship has been found only in females, not in males. And besides behavior, we found that on predator-free islands, lizards with longer hind limb survive better, but on predator islands, lizards with smaller body size, so the shorter SBR, survive better. So now what we would like to know is whether these behavioral and morphological traits could evolve. And to evolve by natural selection, they must affect fitness and they must be heritable. So we already know that they affected the survival fitness over this short period of time of the first experiment, but we are also interested in a longer time scale. So to answer this, we extended the initial study. We increased number of sampled lizards to over 600, and this included 3 generations. So beside the initial sampling that was done after 4 months, now the experimental islands were visited in the next 2 years, 2017, 2018, and summer and fall. And again, the survival was checked, and all new lizards were tagged, and their behavior and morphology was analyzed. And additionally, also we collected tail tissue to extract DNA, and we did the rat sequencing. And this resulted in around 100,000 SNPs that we later used to check population structure, to reconstruct pedigree, and to estimate the heritability of the traits. And additionally, we also analyzed selection over different seasons. So first we did a PCA to look at the population structure of source populations. Here in colors are the 8 source populations, and in gray is the offspring. So in gray circles is F1, and in gray squares are F2s. And there was some structure because 2 of the source population, the ones in blue, were genetically more differentiated from the remainings. And what we can see nicely here is that how F1 and 2 offspring originates from different source parents and is placed between the source genetic clusters. And these mixings of different source ancestry is well depicted by structure results. Here you can see the composition of the experimental islands. In 2016, we found their individuals originating from different source populations. And then in the next years, the mixing of these source ancestry. And for example, when you look at the experimental island 4, you can see that the founders originate from 4 source populations represented by 4 distinct colors. And then in the next years, most offspring originates from the mating of females and males from different source populations. So there are very few bars of just one color. So this is how the genetic material got mixed during the experiment. But then in the next step, we wanted to look at selection pattern over different time periods. So we used GAN models and survival as a measure of fitness, and we estimated directional selection in 3 seasons. So the initial summer, fall 2016, then fall 16, summer 2017, and summer, fall 2017. So the first 2 seasons included only the founder individuals, and the last season included only the next generations of F1s that were born before summer 2017. And this was done separately for females and in red and males in blue, and for islands with predator and without predator. And when we look at forelimb and hind limb, we see that longer limbs are favored in females from predation-free islands. So we have the significant estimates of selection in the first and the last season. And the same trend, but just the trend is observed in predation islands, and the estimates are not significant. What is interesting is that males often show selection direction opposite to the females. And most importantly, we see that for both females and males, the direction and strength of selection is different between seasons. And a similar pattern of selection changes through seasons is observed in case of behavior. And here also, males often show opposite selection direction to females. And exploratory behavior, so the one that is depicted by head-out variable, is important for survival on predation-free islands. So we have this significant directional selection in the first and last season. And on the predation island, time exposed on the ground is under selection, and the lizards that take less risk have better survival. But this is happening only in the initial season. So we find this selection for the traits of interest in different seasons. So then we wanted to check whether these traits are heritable. So we estimated heritability of both morphological and behavioral traits. We used the pedigree that was reconstructed with the rat6 SNPs. and an animal model with mother and island as a random effect, and with the weakly informative parameter expanded priors. So what we found is that heritability for limb length, forelimb and hind limb, is relatively high because over 40%, and also it's high for the long bones of the limbs, so humerus and femur. But we find that heritability for behavior is not significantly different from zero. And this is for both exploratory and the risk-taking behavior. And as behavior usually shows heritability lower than morphological traits, we perform some simulations of heritability to assess whether we have enough power in our dataset and pedigree to estimate significant low heritability values. And we found that we have very little power for heritability lower than 30%. And most heritabilities that are reported for different animal behaviors actually are below 30%. So this could mean that we don't really have power to detect low levels of heritability. However, when using a different method, parent-offspring regression, we see that heritabilities for limbs are very high. So this is represented by the strong relationship between the average parent limb length and offspring limb length. But in case of behavior, there is not even a slight relationship that could point to significant heritability of the two behaviors. So this makes me favor the explanation that simply these two behaviors are not heritable in our Anolis populations. To conclude, selection on both behavioral and morphological traits changes over time. Limb length is highly heritable. Risk-taking and exploratory behavior in Anolis is not heritable or has very low heritability. And evolution of those traits in any particular direction over short periods of time is hard to predict due to the temporal changes in selection, direction, and strength, and also differences between males and females. So thank you. And this is my email in case you would like to discuss some aspects of this work, and I would be happy to answer questions.
I think we have time for a short question. I have a quick question. Could you quickly comment on how your results, do your results have implications for how the lizards would adapt to climate change?
To climate change?
Yeah, or...
Depends what would be the main... Okay, because with a climate change, one thing is we will have more invasive species. So then is the risk of the same risk of facing new behavior. So I would say there is a potential for adaptation, and this, I think this would mostly involve morphology because of high heritability. But yes, there is a potential, but maybe it's difficult to predict exactly how they would do it.
Thank you. Okay, Yasu, you can share your screen and get going when you have everything set up.
Okay, can you see my slide? Okay. Yeah. So, okay, so good evening, everybody, and good morning from Japanese side. So I'm Yasu, Assistant Professor of Ecological Genetics at Hokkaido University. So today I will talk about GWAS of frequency-dependent selection and overyielding in plants. Okay, let's start. So for evolutionary biologists, it is well recognized that frequency-dependent selection can maintain or reduce polymorphism through minority advantage or majority advantage, which are called negative or positive frequency-dependent selection. Another side of frequency-dependent selection is an increase or decrease in population level fitness. For example, our minority advantage or negative frequency dependent selection increases population level fitness at an intermediate frequency of 2 alleles. This dual consequence of frequency dependent selection is theoretically well recognized, but the empirical evidence is very limited. For example, in some cell fry, mixtures of endomorph and gynomorph can increase population level fecundity by frequency dependent reduction of male attacks. However, apart from phenotypic level evidence, genetic architecture of frequency dependent selection and population performance is mostly unknown. Recently, our genome-wide association study, so-called GWAS, provides a powerful tool to study complex traits and fitness components. And GWAS is now extended to interactions among individuals. For example, GWAS of plant-plant interactions about 11 loci are responsible for indirect genetic effects of one genotype on the biomass of other genotypes in Arabidopsis thaliana. And this GWAS also revealed how genotype mixtures outperform monoculture, that is overyielding. But despite accumulating evidence for indirect genetic effect and mixed planting, genetic link between individual and population fitness is still unknown. Okay, in this study, I aimed to connect frequency-dependent selection and overyielding by GWAS. And to achieve it, I applied a recently developed method, GWAS, which is called neighbor GWAS. And previously, we modified selection gradient analysis, in which genotype similarity in a focal locus was considered as an additional covariant. And by calculating genotype similarity at a given locus, this method enabled us to detect the frequency-dependent selection in randomly or interacting genotypes within split populations, or continue the neighborhood interactions in continuous space, like this picture. And in short, this middle regression model shows the self-genotype effect in red and neighbor genotype effect in blue, and self-by-neighbor interactions in green, where genotypes are encoded as +1 or -1 for two homozygotes. And interestingly, coefficient of this self-by-neighbor interactions, that is beta or x by n, determines the slope along genotype similarity, whose coefficient corresponds to the sign of frequency dependent. So further details, please see my previous publication in Evolution. Okay, so to dig deeper into this method, here I showed how regression model represents frequency dependent selection. So here, let us assume sufficiently large population where large A, and small a genotypes are randomly interacting with each other. Then these are numerical examples of 6 representative cases, in which upper and lower rows show the positive and negative sign of self-wide neighbor interaction coefficient, with non-zero coefficients of self and neighbor genotype coefficient. Okay. So among these 6 cases, this red square, case A, highlights the simplest case of symmetric negative frequency dependent selection, where gray and black lines represent the fitness value of large A, and small a genotypes, while the dashed curve indicates weighted mean between genotypes. Okay, so here we see mean fitness increased at intermediate frequency of large A, and genotypes, while larger genotypes had greater fitness than major ones. So this case A means genotype mixture increases population level fitness together with negative frequency dependent selection. And in contrast, the case E shows positive frequency dependent selection, where mean fitness decreased at an intermediate frequency. Okay. And while the other coefficient changes the slope and crossing point of fitness functions, overall pattern remains same. Okay, so now we see negative self-by neighbor interaction coefficient represent negative frequency dependent selection and result in overyielding. So we can repeat this locus level analysis for all SNPs to achieve GWAS and screen significant SNPs with significant negative self-by interaction. Okay, so this GWAS of frequency-dependent selection was applied for paired cultivation data among 98 accessions of Alameda-Vistarian. So Samuel Roost and his colleagues cultivated 10 tested accessions with combinations of 98 genes in a pairwise manner, like this picture. So their data on individual biomass, flowering time, and plant sites were deposited on genome, and it has been analyzed again to detect candidate genes and selection signatures using Arbitraris genome annotation. Okay, so let's go ahead to the result. So here are GWAS Manhattan plots for 3 phenotypes and 3 mutation coefficients, which plotted statistical significance of 3 beta coefficients against 5 chromosomal position in Alameda-Vistarian. So target phenotypes are biomass, flowering time, and rosette size of individual plant, which were analyzed for self genotype or neighbor genotype and self by neighbor genotype interactive effect. Okay. And among these 9 Manhattan plots, I found significant SNPs for self by neighborhood interaction, in which significant QTL was detected on the 5th chromosome. So I focused on this genomic region hereafter. Then I focused on the biomass patterns as the most significant, which shows the negative sign of self by neighbor interactions. By focusing on this significant box plot shows individual biomass in relation to its allelic status at this significant SNP progress. But it's the same as reference in Columbia 0 sequence or alternative. So the left plot shows relationships between individual biomass and the frequency of alternative alleles, in which regression right indicates rare alleles had a greater biomass compared to major ones, that is negative frequency dependent selection. Right box plot shows individual biomass between allelic mixture and or monoculture of 2 alleles at this significance. So here, individual biomass at the mixture was slightly but significantly larger than monoculture, which showed 3% increase in the mixture over monoculture conditions. Okay. Overall, this most significance showed negative frequency dependent selection and overyielding. Okay. So to gain functional insight, then I focused on candidate genes here, the significant SNPs. So SNPs with similar p-values were found. So this QTL region on 5th chromosome was relatively broad, so that it included more than 30 candidate genes. And among them, I found SHATMIN2 SRT2 locus, which is known to regulate other ethylene responsive genes, and thereby affect root growth in Alameda-Vistarian. So this candidate gene highlights the potential role of ethylene-mediated physiological response in relaxed condition and overyielding. But there are too many candidate genes in this region, so I had to narrow down these genes. Okay, so to narrow down candidate genes, I analyzed genomic signature of balancing and directional selection using beta and IHS or EHH score. So these selection genome result of genome scan are shown in the middle and the bottom rows. So near the QTL region, we see 2 relatively strong signals of balancing selection in the middle row, which is beyond 95% of genome-wide spectrum as indicated by this dashed line. So one of the 2 signals showed balancing selection alone, and it did not show directional selection. So the okay, so this signal shows balancing selection alone, but not the signals of positive selection. So going back to GWAS result, we see one candidate gene, SHATMIN2, be located near this signal balancing section. So the combination selection scheme and GWAS suggested balanced polymorphism can underlie frequency-dependent selection and overyielding. Okay, so finally, I'd like to wrap up key findings and discussion points. First, I found quantitative trait progress responsible for negative frequency-dependent selection and overyielding on the fifth chromosome of Arabidopsis. And interestingly, this new QTL overlaps with that is reported by GWAS of indirect genetic effects. So the present study highlights the connection between indirect effects and the maintenance of polymorphisms. And second, I found a candidate gene involved in plant development and root growth, which is consistent with previous evidence that showed root developmental plasticity enabled niche differentiation and overyielding in Arabidopsis. So in conclusion, my study shows the way to connect genetic basis between negative frequency dependent selection and the population level performance. Okay, so that's all for my talk. This work is available as a bioarchive reference. So please check this out if you are interested. Thank you all for listening.
Okay, thank you. Do we have a question we have a question from Brian in the Q&A. I can read it aloud. What does ethylene-mediated root growth mean? Is it ecologically relevant, or is it just they respond with these loci if we dump ethylene on them? Sorry, I don't know Arabidopsis well.
So because I can see the message in the chat, so that.
Oh, it's in the Q&A, which is separate from the chat, but I can also.
Q&A, okay. Q&A, okay, yeah. Yeah. So still, this one is just a candidate gene. So I'm not sure this gene how this gene play a key role in the competition. But the previous study by Samuel Roost et al. Showed the root plasticity enabled so the below ground differentiation, niche differentiation, and enabled overyielding in Arabidopsis thaliana between two genotypes. So there, so maybe so the equivalent is one of them to achieve root plasticity. So that this kind of so the morphological differentiation between two genotypes or accessions might result in overyielding within small body, so that to allow them to packing in a small body and so on. But this is just a speculation.
Mm-hmm. Yeah, this was a great talk.
Well, if there are no more questions, this is a somewhat shorter session. We originally had 4 people scheduled. On Wednesday, 2 of them withdrew, so we're down to only 2 people in this final session. This concludes our virtual evolution, so thank you all for your time and attention. A big thank you in particular to the moderators and the volunteers who made this all much more possible, other than our ability to get things done. And for those of you who will be joining us in person, we will see you in a month in Athens, Georgia.